All right, great. It's a lot of fun to participate in this meeting. It's marvelous to hear about all this recent data, and I think especially what Lauren talked about fits in well with some of the stuff, as he mentioned, that Mark Brandon did in my lab a little while ago. So I added that into the middle of my talk specifically because of that connection. But in general, we've been very interested in mechanisms for episodic memory in the hippocampus and entorhinal cortex, and in particular, as you've already kind of heard, coding of episodic memory requires knowing the location and the time of specific events for the episode that's being stored. And so we're very interested in the mechanisms for coding these, the space and time of events that seem to involve circuits in entorhinal cortex and hippocampus. And there seems to be important roles of different inputs to the entorhinal cortex in the mechanism for coding of space and time. So we've done work on the influence of the medial septum on the entorhinal cortex, but then there's also data that I'll talk about at the end that implicate the important role of cortical sensory input coming into these regions from areas such as retrosplenial cortex. So here's an overview. First, I'll talk about some of these medial septal inputs, which connects with what people like Lauren have talked about in terms of the theta, the role of the theta rhythm for coding of the spatial representation in grid cells, as well as the coding of time. Then I'll talk about the alternation, which we call theta cycle skipping, and then I'll talk about visual coding. So just to kind of give an intro for the grid cells, this is a movie of a grid cell recording from our lab. As you know, grid cells were discovered in the lab of Edvard and May-Britt Moser. The paradigm that's commonly used for studying grid cells is to have a rat foraging in an open field environment, and you can track its head direction and location based on LEDs attached to the head. And you can see the black line is showing the trajectory of the rat, and the red dots are showing the location of the rat each time an individual cell fires a spike. Obviously, that video has been sped up quite a bit. And you get these responses that are falling in a regular array of locations in the environment, falling on the vertices of tightly packed equilateral triangles. Now, right up front, I want to mention that there is interesting data implicating a role for theta rhythm in grid cell firing. We don't fully know the mechanism of its role in grid cell firing, but I want to emphasize that the Moser lab showed very strong relationships of the spiking of grid cells to the ongoing theta rhythm in the entorhinal cortex. And here's an example that the cells show a phenomenon of theta phase precession, where when the rat first enters the firing field of a grid cell, the spikes occur at the end of the theta phase, in theta cycle, and move to earlier and earlier phases, which is very similar to the theta sequence retrieval that Lauren talked about, and that was from David's paper. But David's actually shown that they're actually distinct. You can have one without the other. But you see this very clear phase precession in each of the firing fields. And it's also been shown, this is data from that Jason Clymer did in my lab, where we had the rats running through two-dimensional firing fields, and he still saw theta phase precession for grid cells. And this connects with data that we had a number of years ago from Mark Brandon, showing that manipulations that affect theta rhythm affect the spatial periodicity of grid cells. So he had recordings of theta rhythm in the baseline condition during recordings of nice spatial periodicity in individual entorhinal grid cells. And then he performed inactivation of the medial septum with the GABA agonist mucimol and saw a loss of the theta periodicity and the loss of the spatial periodicity of the grid cells. And then as the theta frequency comes back, you see a regaining of the spatial periodicity of the grid cells. Now, in addition, in more recent work in collaboration with Howard Eichenbaum's lab, Mark Brandon and Ben Krause, and in this collaboration, showed that the grid cells will also fire as time cells, which is a term that Howard coined. So this shows the individual grid cells that was already characterized as in a two-dimensional open field. Now it's being recorded as a rat runs on a treadmill, and you can see that there's silent for a period of time, then it fires for a period of time, then it's silent, and it'll fire at the end of the running. All right, and then you'll see it start another trial. A little firing right at the beginning, stops, fires again for a period of time, then stops, fires again for a period of time. So this is corresponding to something like this. It's a different cell, but it's the same sort of phenomenon where the cell has been characterized as a grid cell in the open field, And then on multiple different trials of running on the treadmill, it fires at the beginning, then it stops firing, then it fires again, then it stops firing, then it fires again. So it's a very striking characteristic. I want to emphasize that these time cells were initially described in hippocampus, but this is showing that the cells that fire as grid cells will also fire as time cells. Is that a question?
Yeah, so why are you saying time cells? And because, I mean, the rat or the mouse is still moving, right? So yes. I just want to understand that.
Yeah, so that's actually, so it's a good point, and I didn't have time. I took out a lot of slides that I normally show, but we actually did an experiment actually in this paper with Ben Krause in 2013, where we specifically looked at different treadmill speed so that we could dissociate time of running from distance of running on the treadmill. And it actually turns out that some cells will code distance of running, but some cells will code the time duration of the running. So yeah, I wish I still had that slide in there. And just to connect to the data on grid cells, just as grid cells will lose their spatial periodicity with medial septum inactivation, Eva Pastelkova showed that time cells, in this case in hippocampus, will lose their time specificity during the medial septum inactivation. So here's a time cell firing at one time and then completely shuts off. Here's one that's firing a couple of different times and it keeps firing but loses its specificity. And here's a whole array of different time cells, and you can see that they lose their temporal specificity during the medial septum inactivation. So this is really exciting in the sense that both the spatial coding and the temporal coding seem to involve similar mechanisms dependent on the medial septum, but we don't yet know those mechanisms. There's a number of inputs, GABAergic, cholinergic, and we're still trying to differentiate these different inputs from medial septum. Now there's a number of models of grid cells, and I should say right up front that actually all these models can account for time cell type responses as well. So there's the attractor dynamic models of grid cells that can integrate velocity, speed, and direction, but they can also, if you assume there's some kind of steady input, they can also give the movement through time. There's also the oscillatory interference models, which can very effectively, as shown here, simulate the spatial periodicity of grid cells, but then also can very effectively simulate the multiple firing fields that I just showed you during the running on a treadmill. So both of these types of models are potentially able to address this data, and I guess I really want to emphasize these types of models because we still haven't really resolved fully the details of the mechanisms of these models. In particular, one thing I mentioned in this slide is that the attractor models are consistent with a lot of the data, but they have not yet simulated theta phase precession in two-dimensional environments or in temporal coding. They've been shown in the McNaughton lab that can do just one-dimensional environments. So I think, I mean, the dominant feeling in the field is, and I agree with this, the attractor dynamics are important for grid cell firing, but we really have a number of different questions that need to be resolved, particularly how you get this theta phase coding. And as I mentioned here, there's conflict between this model and the fact that the data from bats, such as that gathered by Michael Yartsev, it can show grid cells without theta rhythmicity. So that's a big question mark for this model. But moving on, I want to talk about the fact that both these types of models integrate velocity for generating the grid cell responses. So they use both direction and speed, and this is consistent with other types of neuronal responses in the entorhinal cortex. In particular, there's cells that code running speed as animals are moving around in an open field environment. These will change their firing rate with the running speed of the animal, and there are neurons that code head direction, originally described by Jim Ronk and also by Jeff Taube. So in recent work that Jake Hinman and Mark Brandon did in my laboratory, we've actually looked at these different types of coding of running speed. A number of different groups have shown influences of running speed on the firing properties of neurons. In particular, all of the different cell types in the entorhinal cortex actually will change their firing rate, or at least examples of them will change their firing rate with the running speed of the animal. So grid cells, conjunctive grid by head direction cells, and then what the Kropf paper from the Moser lab called speed cells, which are cells that are usually interneurons that didn't have any other kind of functional coding, but show very clear coding of running speed. But in this paper, we were focusing on the fact that in addition to the firing rate change with running speed, you have a change in theta rhythmicity with running speed. So Theta rhythmicity can be detected by doing an autocorrelogram. So if you actually take the spike train relative to itself and they're firing at about 8 hertz, then they're going to start to line up as you shift, to line up again at 125 and then 250 milliseconds. So these peaks reflect the 8 hertz periodicity of firing. And as the rat's running at low speeds, you have about 8 hertz periodicity, and then it'll actually, you can see these peaks get closer together as the periodicity increases in frequency with higher running speeds. And this is shown in a number of different cell types here. So it seems like there's actually 2 different ways of coding the running speed, and they seem to be differentially sensitive. So when Mark Brandon did the medial septum inactivation, in some cases he completely lost the theta rhythmicity of the spiking of the neurons. But even though they lost their theta rhythmicity, they would actually still show very clear firing rate changes with running speed. In fact, they actually show better linear relationships to running speed compared to more saturation here. And in cases where he had medial septum inactivation, but he didn't completely wipe out theta rhythmicity, the important thing to show here is that, again, the firing rate change with running speed is maintained, but you actually see a change in the relationship of the theta rhythmicity to running speed. Here it's getting higher frequency rhythmicity with higher running speed. Here it does not maintain that relationship. So this suggests the interesting idea that maybe the loss of the grid cell spatial periodicity with medial septum inactivation is somehow due to a loss of the speed modulation of spiking rhythmicity in these neurons, which is a potential role for theta rhythmicity in the mechanism of grid cell generation. All right, so I wanted to mention that all of these models require movement direction as their input, and they cite papers about head direction, but you may have already kind of thought about the fact that head direction isn't necessarily movement direction in the environment. And we explicitly just looked at this in a paper. We looked at the behavior of the animals in the environment and showed that the head direction does not always line up with the movement direction. You can imagine that as you're walking down a street, you can be looking to one side or the other differentially for different amounts of time. And we actually tested during periods where they don't line up, tested to see whether cells were coding head direction or movement direction, and found that there were no cells that were purely coding movement direction, even though, as in previous studies, many cells show coding of head direction, and only a small number show coding of head direction and movement direction. So that really suggests the dominant code is for head direction, which is an interesting question for these types of models. Why is it that the head direction is predominantly encoded? And I want to emphasize both of the types of models I described, the attractor models and the oscillatory interference model, will show great coding if you give movement direction as input, but their coding will break down if you give neurons coding head direction as the input to the model. All right, so this raises these interesting questions. What's the role of theta rhythm in the generation of grid cells and time cells, and why is head direction coded more strongly than movement direction? And in some ways it might seem obvious that maybe it's because sensory input is more important for coding of grid cells than path integration is. All right, so now I want to kind of go to this digression that I added to talk about in relation to Lauren Frank's data, because it's really very exciting to me. This is something that I actually, we saw a number of years ago, and I was very interested specifically in the possibility of this sort of trajectory retrieval. So it's really exciting to see what Lauren's shown. I'm not going to show you location representations of different phases. We're going to focus on head direction representations. But as Lauren mentioned, in the entorhinal cortex, initially Kate Jeffrey showed what she called theta cycle skipping, and then Sachin Deshmukh and Jim Knierim's lab showed this, and then it's also been shown in the medial septum. This is, you can see this with the autocorrelograms, and you saw this in the cross-correlograms that Lauren showed. If you have a standard autocorrelogram, it has a peak at 125 milliseconds, indicating 8 hertz rhythmicity, but if you get this theta cycle skipping, you have a small peak at 125, and then a larger peak at 250 milliseconds, indicating that on average they're tending to fire every other cycle. And here's an example of that, where the cell is spiking on one cycle, then it's quiet for one theta cycle, then it spikes, and it's quiet, then it spikes. So exactly what Lauren showed to you. But in our case, we were primarily seeing this in head direction cells, and what we did was to look at different head direction cells in terms of their cross correlations. So we had 2 examples of 2 theta skipping cells shown by the autocorrelograms, and then looked at the cross-correlograms, and just as Lauren showed for 16% of the cells, here in this case these cells are firing on the same cycle and then less often on directly alternating cycles. So here you can see they're firing together, then they're off, then they're together. But then just like Lauren showed, and this is really marvelous to see, to see the convergence of this data in cross-correlations of pairs, so theta cycle skipping pairs, we saw examples just like he saw where there was a zero cross correlation at 0 milliseconds and then a nice strong cross correlation at 125 milliseconds. And then this is just an example. I mean, it's not always this clean, but in this case, you can see these cells are firing, you know, green on one cycle, then blue, then green, then blue, then green, then blue, just like Lauren was talking about. So it's really a beautiful association. But as I mentioned, it's primarily in head direction. This is recording in entorhinal cortex rather than hippocampus. We didn't have that many grid cells, you know, certainly not recorded simultaneously, so we couldn't do the sequence prediction or the sequence decoding, but we could look at the head direction cell coding, and you can see when they're synchronous, we actually often saw many cells that shared head direction tuning. But for these anti-synchronous pairs, we saw no pairs that had head direction tuning closer than 40 degrees from each other. Now, obviously, they had to have some overlap in their coding, or they wouldn't be spiking together. All right, but what we saw was that cells that were anti-synchronous tend to have head direction coding that was further apart. And as Lauren beautifully showed, you know, with your angular velocity, the rat can't zip their head back and forth at theta rhythm. You know, try that. You'll get a really painful neck if you try to move your head that quickly. And even with the small heads, they're not doing that, as he showed. So it really isn't that they're moving back and forth between. What it seems like is that if they're, you know, in a particular orientation where they could activate two head directions, they seem to maybe be switching back and forth between these two possible head directions and potentially doing trajectory coding, though we don't know that from our own data, but Lauren's data suggests that. So this was very exciting to me because actually earlier on I'd done simulations where we'd shown, you know, separation of encoding and retrieval on different dynamics, and related to this, discussed the idea that encoding would be driven by the current sensory input and the retrieval would be on a different phase of theta, which is necessary to get nice, clean representations to avoid the sort of interference that Lorenz actually shows can happen in these types of models. And so in this paper with Howard Eichenbaum, we actually talked about how the retrieval could be happening on a particular phase of theta. It also relates to work we did that Murat Erdem did in my laboratory, where we were showing how grid cell representations could do forward trajectory retrieval in multiple different directions to do kind of maybe a model-based sampling of potential trajectories for goal locations, similar to what David was talking about. All right, so in the last section, I want to kind of get back to talking about the potential cortical sensory influences on grid cells. And as I already mentioned, this predominance of head direction selectivity in entorhinal cortex suggests an important role of, you know, head direction is important for determining the angle of sensory input. So there's a lot of data indicating a role of visual sensory input on grid cell firing. So initially, Caswell-Berry showed this effect where if you moved the barriers in an environment, it would actually cause a compression of the spacing of grid cell firing fields. And then this was replicated by the Stansulas in the Moser laboratory. And they showed this additional characteristic that different grid cell modules seem to respond differentially to barriers. So in this case, they showed grid cells that had a narrow spacing between the firing fields, usually recorded in more dorsal regions, actually didn't respond to the barrier compression. So these firing fields have about the same distance, even with the border movement. Whereas grid cells in more ventral regions with larger spacing between the firing fields, when they move this barrier of the environment to make the environment smaller, the actual spacing between the grid cells compresses in that direction. So with Florian Raudi's, we actually modeled how this could arise from the angle of visual features or optic flow. And really, you can use either thing for coding location. I made this movie with these random dots just to show how easy it is to get a sense of your location in an environment just based on either optic flow or feature angle. And we looked at the ground plane characteristics using template matching from the Perron technique or features on the walls, so visual feature angle. And we showed that we could separately simulate, you know, obviously when you're focusing on features on the ground plane, you don't see an effect of the movement of the walls, consistent with the, you know, the lack of the rescaling in these smaller firing fields or smaller spacing firing fields. Whereas when the spacing was larger, we modeled this as the network responding to the features on the wall, and when we move the walls, then we get compression of the spacing between the firing fields. And so it's kind of intriguing because these neurons are more in dorsal, these are more in ventral, and if you think about it, this is being projected, ground plane is going to be more projected on the dorsal retina. Walls will be more on the ventral retina. So it could be that there's some retinotopy in the medial entorhinal cortex, which I've been scouring the literature to see if it's in any of Callaway's data. There's tons of visual areas on the dorsal surface, but unfortunately, entorhinal cortex is on the side of the brain, and they don't get down there and to do the intrinsic imaging. But connected to this now, Florian had used his same techniques of the optic flow to actually model what Neil Burgess proposed as allocentric boundary cells. Neil originally proposed these way back in 1996 based on stretching of the environment or compressing of the environment, the effects on place cells. If the environment is stretched, place cells will often expand, and he proposed that this could be due to boundary cells that are responding to specific barriers in the environment. And many years later, after this very, very clear prediction, many years later these cells were described, where there would be cells that would respond consistently to one allocentric boundary in the environment. In this case, the west wall is causing responses, or here the south wall is causing response. And if they insert a wall, you see responses against that side of the inserted wall. You see the same response against the edge of a tabletop, and when they pull the tables apart, you see the response at the gap between the tables. So it indicates that these boundary responses can depend not only on a clear visual wall, but also can respond to a drop-off that the animal knows is essentially a boundary on its environment. So as I mentioned, Florian Rowdy's used the same template matching for optic flow to detect the boundary between the ground plane and the wall, or between a ground plane and a distance wall when there's a tabletop. Now, Neil Burgess really has been very prescient in his descriptions of these networks, because he not only proposed these boundary cells well before they were discovered, but he also proposed that they could arise from an interaction of egocentric representations of features with head direction cells, and proposed that they could converge in areas such as retrosplenial cortex. And so he was proposing not only allocentric boundary cells, but also egocentric responses to boundaries, and plotted this in this paper. And more recently, Jake Hinman has shown these egocentric boundary cells that Neil Burgess predicted. Now, interestingly, Jake Hinman was recording in the dorsal striatum. We don't normally record in striatum. He was initially aiming for lateral septum and hit dorsal striatum and saw these very interesting cells there. But I mentioned in the context of retrosplenial cortex because now Andy Alexander in my lab has shown them in retrosplenial, and he's shown that they give, and we know that the area of dorsal striatum is actually receiving input from retrosplenial. So this shows examples of these egocentric boundary cells. They're similar to something that Dori Durdekman also described, where when the rat's running around in the environment, this particular cell is responding when he's going east on the south wall, and when he's going west against the north wall, which if you think about it corresponds to having a wall on the right side, the egocentric right side of the animal, which is plotted here in polar coordinates, where that's front of the animal, this is right. There's also cells that respond when the wall is on the left side of the animal. So there's this coding of barriers relative to animals' egocentric perspective. And the way this is coded here, you can see that cells responding with spikes under different barriers, and for each spike then Jake plotted the location of the barrier, and with multiple barriers then you get a sense, oh, this cell is tending to respond when there's a barrier at about 10 to 20 centimeters in the front right of the animal, and he does occupancy normalization to see that across the mean occupancy. And he can dissociate the egocentric and the allocentric reference frame. So he also saw head direction cells in the striatum, which have been described by people such as Sherry Mizumori. And he showed that these allocentric head direction cells will respond based on the overall environment, even if he rotates the local environment. But when he rotates the local environment by 45 degrees, the egocentric boundary cells maintain their egocentric representation. So the actual responses will rotate with the environment. He also showed that the responses are stable across multiple different environments, so it's not that it's remapping the way place cells would. And he showed, similar to what the Burgess lab and others had shown, here you see a response for an egocentric cell against a wall, here's a response for a tabletop. So they seem to be responding not just to simple visual features, but to the presence of a barrier in the environment. So this fits very well with this framework that Neil Burgess had proposed for this coding of egocentric representations that could create the allocentric representations, both of the boundaries and ultimately of place in the environment. All right, so I just wanted to kind of leave you with some of these interesting questions. We still don't really know. I think there needs to be further modeling work, even with the attractor models, to address how theta rhythm is influencing the response characteristics in these circuits. Why is head direction coded more strongly to movement direction? And how do neural circuits code position and self-motion from visual feature, angle, or optic flow? We've used relatively abstracted models for that, and I'd like to see them be more biological. And finally, I want to give credit to Jake Hinman, who did the egocentric boundary cells, and Mark Brandon, who did the work on the medial septum inactivation, and Florian Raudi's, who did a lot of the modeling I talk about. Thanks.
Michael.
I had a question about the visual aspect of it. So why do you think it's necessarily just visual, rather than, for example, using the whisking?
Yeah, we have thought about that, and I didn't mean to imply that there couldn't be. In fact, I said sensory cues at the beginning, because it is very possible, especially for the egocentric boundary cells that are responding quite close to the animal, that the whisker input could be significant. But there certainly were cells, or cells that were responding to barriers at 10 or 20 centimeters where it was beyond the range of the whiskers.
Yeah, I hadn't thought about the head direction versus movement direction thing. It's incredibly striking to me that everything falls apart when you use the signal that's actually there. So do you have any ideas about how that works? Because I'm at a complete loss.
Yeah, I mean, my best idea is really that the animals are probably using it, that it's more important to know the head direction, translate the visual cues from the retinotopic coordinates to allocentric coordinates, and then do what roboticists call visual odometry, to get running speed, to get speed and direction from the visual sensory inputs rather than a proprioceptive input.
So if this were true, there should be a difference between the red moving around in dark and in light, right? Maybe this might be an experimental prediction that if it's in dark, maybe it's the body direction that's more dominant, and in light it's more head direction.
Yeah, I mean, the head direction cells will fire in darkness still, and there's but people have not specifically tried to find movement direction cells in the darkness. That's actually an interesting point, and we have started to look for movement direction cells now to see if they would show up.
But if the brain was doing that computation, then wouldn't you see a signal for like the output of that?
Yeah, you would expect that you'd see that signal somewhere. And I mean, when we actually were trying to, we published this paper in a special issue in Brain Research, which is, that's where, John O'Keefe first published grid cells, or place cells, but Brain Research wasn't our first journal of choice, and that's kind of exactly the argument that people were making. They were saying, well, these movement direction cells have to be somewhere, and you haven't proven that they're not somewhere else, and that's very true.
Do you want to predict that maybe the head direction skipping is driving the theta sequence skipping? Because in Lauren's talk, the directionality difference and the upcoming decision difference were all corresponding to different directions, possible head directions. I'm thinking, would you predict that in, let's say, David Reddish's task, where the animal has to continue along the same path but for different lengths, get to different goals, that if there's some kind of, that there wouldn't be a data skipping effect there because the head direction signal doesn't differentiate.
Yeah, I mean, I guess that would be an interesting prediction. I mean, we were recording in an open field environment where there certainly were many options for directions of going, whereas in his task, you were talking about different lengths along the same circle track, and so you wouldn't necessarily differentiate them. Though what you would predict is that there ought to be a systematic change maybe in head direction as it's reading out a trajectory that goes through this kind of curved change in head direction.
Make a prediction.
Yeah, that'd be great.
Egocentric boundary cells are interesting. We don't normally think of dorsal striatum as being a way station on the route to allocentric computation. I wonder if there's an alternative explanation, for example, signaling whether it's a good idea to move to your right or something like that.
Yeah, so I think you're right that, and I have kind of thought about the fact that, yeah, if you're, navigating in an environment and making choices about actions, you need to know that there's a wall on your right so you don't bonk your head into it. We've all experienced that. So I think that's a good point of the functional role of that output. But in terms of the Neil Burgess model, I was thinking more those are reflective of the input from retrosplenial.
So the differential expansion of the grids at different scales when an environment is resized, I think of that result as also related to the result of expansion of all grid fields in novel enclosures. And I was wondering if you know whether in a novel enclosure there's a differential expansion of the ventral versus dorsal fields.
Yeah, so they didn't have as many modules. I mean, that Stensrud study was very heroic in terms of getting, simultaneous recording of 4 different modules. And the Berry papers, in addition to the barrier movement, they also did this in novel environment, but they never had more than 2 spatial scales at one time. And Caswell said they don't see that differential compression even when they did the compression in their manipulations. So that's an interesting puzzle there.
There's an alternative interpretation of that scaling effect with the boundary compression and differences. And it's actually something Francesco Savelli and I had written a little bit about. And there was a poster from Kevin Kynath and Russell Epstein's lab at SFM where they did modeling, where they replicate that effect not by actually compressing the grid, but what happens is as the animal moves closer to the wall, which is not where it's expected to do, it causes a shift of the grid as the boundary corrects the proper phase of the grid. Mm-hmm. And if the amount that the boundary went was more or less the same period as a grid, then there is no correction to be made, and it looks like it's fine. But if the scale is bigger, and then when the grid shifts to correct its phase, it appears as if it's a compression of the whole grid. So there's and it is modeling which actually seemed to the extent that I had the poster, looked pretty decent. So it's actually an interesting kind of thing, whether that effect really is compressing the grid versus really just being an indication of another thought that these boundary cells are supposed to do, is to actually be a way to reset the phase of the grid. And Lisa Giacomo, of course, she actually has a paper kind of showing that. So it's actually, they're alternative.
Yeah, we certainly, this is just one hypothesis. There's certainly, that's very plausible that you have that resetting.
Okay, well, we should probably move on. We have a break now, and we'll resume at 3:30.